Greetings. I hope you're all enjoying the symposium today. I'm presenting published results from field studies with the Sagebrush Step Treatment Evaluation Project, or SAGE-STEP, and some preliminary results from studies at Grand Staircase-Escalante National Monument. This work addresses the ecohydrologic and erosional impacts of pinyon and juniper encroachment and tree removal on sagebrush rangelands. I have a number of co-authors acknowledged here, most of which are with or were with the USDA-Agricultural Research Service when we did this work, and also Raven Chavez from the BLM, our collaborator on the Grand Staircase-Escalante National Monument studies. The photos here show the typical structure of a sagebrush rangeland and pinyon juniper or PJ woodland. We know the vegetation and ground cover structure on these landscapes strongly regulates surface hydrology and erosion processes. For sagebrush rangelands, interspace microsites between shrubs, as shown in the photographs to the left, are the primary sources for runoff and sediment. But these interspace microsites are isolated such that runoff and detached sediment travel only short distances downslope. For woodlands, as shown on the right, inner canopy areas comprised of shrub and interspace microsites are the primary sources for runoff and sediment. Downslope transfers of water and sediment are regulated by bare area connectivity and formation of concentrated flow throughout the intercanopy. For both these systems, runoff and erosion responses for a given storm then are largely controlled by the spatial accumulation of runoff and sediment and by process connectivity. Therefore, treatments targeting enhanced hydrologic function should aim to increase cover, reduce bare area, and thereby limit runoff and sediment sources to process connectivity. The aim of the work I'm presenting today is to populate this conceptual model with quantitative data. The work I'm presenting addresses two primary research questions as shown here. First, what are the impacts of PJ woodland encroachment, or woodland encroachment for short, on vegetation, soil properties, and hydrology and erosion processes? And second, what are the impacts of tree removal on woodland encroached sagebrush rangelands? I present results from two ongoing projects. First, I'll present published findings from the Sagebrush Step Treatment Evaluation Project, or SAGE-STEP hydrology study. And second, I present preliminary findings from ongoing research at the Grand Staircase-Escalante National Monument in partnership with the BLM. I'll provide a very brief overview of our methodology here, but for those that are interested, reference the QR code for more specific details. We utilize a suite of infiltrometer, rainfall simulation, and overland flow experiments to quantify surface hydrology and erosion processes. The various methods allow us to isolate specific processes that occur at fine, coarse, and hill slope spatial scales and provide data for representing those processes in hill slope scale conceptual and quantitative models. Infiltrometer methods allow us to isolate the effects of soil properties, whereas the rainfall simulation and overland flow experiments incorporate the effects of vegetation, ground cover, and soils on hydrology and erosion processes. In addition to the hydrology and erosion experiments, we quantify a number of vegetation, surface conditions, and soil attributes at each site to determine the controls on hydrology and erosion processes. These include, but are not limited to, foliar and ground cover, plant density, soil characterization, soil physical and hydrologic properties, ground surface roughness, soil stability, and litter depth. Since the early 2000s, we've been conducting the aforementioned field experiments at multiple sites spanning the Great Basin region, as shown on the map to the left. Sites noted with red symbols are part of the SAGE-STEP study, and locations noted with blue symbols indicate additional study sites. The study sites span a range of vegetation types, conditions, and soils, as noted to the right, and include repeated studies on untreated sites and on treated sites in various time steps following tree removal by fire and mechanical methods. This work is well replicated, as you can see by the numbers to the left, and the respective database is perhaps the largest of this type in the literature. We will now take a quick look at some of the data from the SAGE-STEP study. As you can see in the graph to the upper right, cumulative runoff and erosion, represented as cumulative sediment yield, are linearly related for these sparsely vegetated communities, and runoff and erosion both increase exponentially where bare ground exceeds 50 to 60%, as indicated in the graphs to the lower right. This occurs because runoff and erosion sources and processes are well connected, and concentrated flow is the dominant erosion process under these conditions. The photographs on the left show pre-fire and post-fire conditions at two woodlands. We find burning tends to homogenize runoff and sediment sources on these landscapes through cover reductions and thereby increases runoff and erosion potential the first few years post-fire, as you can see by the red bars in the graph to the right. However, cover increases over time, as can be seen in the photographs, and those cover improvements tend to reduce runoff and sediment delivery as indicated by the green bars in the graph to the right. Such changes are particularly desired for previously bare intercanopy areas in order to limit runoff generation and prevent process connectivity. For mechanical treatments, as shown here, initial impacts depend largely on how well the treatments disperse tree debris into bare patches. Over time, residual tree debris and improved cover conditions limit runoff and erosion, as reflected in the blue and green bars in the graph to the right. However, our work has found that post-treatment reductions in runoff and erosion occur earlier and are greater for burned versus mechanical treatments such as cutting and mastication. There are, of course, caveats to assessing benefits of fire versus mechanical treatments. We have found that fire tends to rapidly improve perennial herbaceous cover and hydrologic function, but at the expense of sagebrush reduction and increased cheatgrass in areas previously underneath tree canopies, as can be seen in the photographs to the left. Mechanical treatments in our research effectively removed trees, retained sagebrush, and resulted in less cheatgrass cover, but the improved hydrologic function was delayed for mechanical treatments relative to that following burning. Also, we have found, as you can see in the photograph to the right, that mechanical treatments tend to leave numerous small trees to reestablish woodland conditions over time, necessitating follow-up treatment. Field studies from SageStep have provided a basis for a new framework to model the hydrologic and erosion impacts of woodland encroachment and tree removal on sagebrush rangelands. We recently published a framework for such applications of the RIM-Web tool. The study demonstrated application of RIM in assessing hill slope runoff and erosion processes on untreated woodlands and predicting hydrologic and erosion responses following various tree removal practices. The model can be used to assess current conditions, predict potential impacts of treatments, and targeting management practices. Our ongoing work with SageStep includes continuing to follow treatment responses at the hydrology sites indicated with yellow symbols on the map, and to utilize our recent model advances to assess site long-term responses to treatments across the full SageStep woodland network indicated by both the red and yellow symbols on the map. We will now shift gears and discuss preliminary results for ongoing projects at Grand Staircase-Escalante National Monument, including assessing the ecohydrologic impacts of woodland encroachment and tree removal by mastication, and experiments assessing the effects of biocrust cover on hydrology and erosion processes. This work is in direct collaboration with Raven Chavez of the BLM. For the first study, we conducted experiments at a sagebrush-dominated site and an adjacent PJ-dominated site to assess the impacts of woodland encroachment. The respective plant communities reflect two distinct ecological states on the same ecological site as indicated here. The sagebrush site has a fine vegetation structure with ample shrub cover and limited bare interspace area. The woodland site has a coarse vegetation structure of tree islands representing 30% of the site, and then also has numerous dead shrubs, sparse understory cover, and extensive inner canopy area with variable bare soil and biocrust cover. The photographs here show conditions across the various microsites at both sites, and I'll summarize here. For the sagebrush site, interspaces occupy about 70% of the area and are sparsely vegetated with moderate soil stability. Sagebrush microsites occupy nearly 30% of the area and are well vegetated and have moderate soil stability. For the woodland, interspaces occupy about 70% of the area and are essentially bare with the exception of biocrust cover. The biocrust cover provides good aggregate stability where it occurs. Shrubs are sparse throughout the woodland, occupying less than 1% of the area, and under canopy areas are mostly bare. Tree microsites occupy about 30% of the woodland area and are 100% covered by the variable thickness litter layer underlain by hydrophobic soils with generally lower than expected aggregate stability. The data shown here are for rainfall simulations on 0.5 meter square plots. The graph on the left shows the infiltration rate and the graph on the right shows the sediment discharge rate for the various microsites at the sagebrush and woodland sites. You can see that infiltration was lowest and sediment discharge greatest for the interspaces at both sites, shown as the red and orange lines on the graphs, and that the shrub microsites, shown as black and blue lines, have the highest infiltration and the lowest sediment discharge. So for both sites, the interspaces are clearly the primary sources for runoff and sediment at the fine spatial scale. Note infiltration is generally low for tree microsites due to water repellency, but individual trees were removed for placement of the rainfall simulator. If trees were left in place, tree interception would partially buffer repellency effects at the ground surface. Lastly, note that interspaces occur over about 70% of the area at both sites and are spatially well connected solely at the woodland site. Starting on the left and progressing to the right, the graphs here show that sediment yield increases linearly with runoff for the interspaces shown as red and orange symbols, and that as shown in the other two graphs, both cumulative runoff and sediment yield are reasonably well correlated with percent bare soil. The variability in both runoff and sediment yield for woodland interspaces is associated with variation in biocrust cover, which I'll address in a subsequent slide. The high levels of runoff for well-covered tree plots, shown by the green symbols, is associated with strongly hydrophobic soils underneath the litter layer. Here again, we have graphs showing infiltration on the left and sediment discharge on the right. For these graphs, the data are restricted to woodland interspaces only and show separate curves for low crust cover in red, moderate crust cover in blue, and high crust cover in green. You can see that infiltration was lowest and sediment discharge the highest for plots with biocrust cover less than 20%, and that moderate biocrust cover, which is crust cover 20 to 50%, yielded the highest infiltration rates and the lowest sediment discharge. Our hypothesis here is that the lesser infiltration and higher sediment yields for high versus low crust cover is associated with poor clogging. A follow-up study is underway to address this hypothesis through more extensive replication across varying crust cover. The photographs here show cover and surface conditions on 12 square meter plots at the sagebrush site, as shown to the left, and on intercanopy and tree plots at the woodland site, as shown on the right. In summary, the sagebrush site was well vegetated with shrubs and had limited bare ground with shorter gaps between plant bases and canopies in comparison with the woodland. For the woodland, intercanopies had only 6% foliar cover and were largely bare at ground level, with exception of biocrust. These microsites were well connected throughout the woodland. Woodland tree microsites had limited foliar cover, but had more than 80% litter cover over hydrophobic soils. Plant basal and canopy gaps were extensive throughout the woodland. Here we have data on runoff and sediment variables for rainfall simulations and overland flow experiments at the sagebrush site and in intercanopy and tree microsites at the woodland site. Runoff, sediment yield, and overland flow velocity were highest for the woodland intercanopy plots shown in the middle of the slide. These areas are, of course, the primary sources of runoff and sediment at the woodland, and these areas are more roadable than the hill slopes at the sagebrush site. That's attributable to the well-connected bare patches throughout the intercanopy. Flow paths were generally wider at the sagebrush site, as shown on the left, with flow routing around obstacles and at a lower velocity than measured for intercanopy plots. Runoff and sediment yield were higher than expected for the tree plots, largely due to hydrophobic soils. Water tended to pond in the low areas, as shown on the right, and then the draining of those ponds was sufficient to move litter and expose dry surface soil to runoff where it occurred. Overall, the preliminary results for the sagebrush to woodland comparisons are consistent with conceptual models of these systems. Runoff and erosion rates are greater for the woodland due to the coarsening of the vegetation structure with woodland encroachment. Runoff from interspaces accumulates through well-connected bare intercanopy areas and forms high velocity concentrated overland flow with ample energy for sediment detachment and transport. The intercanopy throughout the woodland clearly depicts evidence of these connected processes naturally occurring during runoff events, as shown in the photographs here. We are continuing to examine the effects of crust cover on surface processes and the impacts of site-level tree removal in woodlands. The data shown here are very preliminary, but give us a good first look at variation in surface conditions across early to late successional stages of biocrust development for the woodland intercanopy. In general, the preliminary data suggests that areas with more than 40% dark biocrust cover exhibit a rougher surface, greater aggregate stability, and delayed time to ponding and runoff generation. Parsing the various effects and that of bulk density on overall hydrologic response and erosion potential is forthcoming. This well-replicated study will provide a basis for assessing the aforementioned hydrologic and erosion responses across low, moderate, and high biocrust cover. The results summary here is also very preliminary and includes some basic field observations for our ongoing work assessing the ecohydrologic impacts of a mastication treatment with seeding. The study will compare a suite of vegetation, soil hydrology, and erosion response variables across the sagebrush, woodland, and mastication study sites to assess treatment effects. All three sites are on the same ecological site. Vegetation and surface conditions at the mastication site were similar to the woodland site prior to treatment. The preliminary data suggests that after a 10-year period following tree removal and seeding, the mastication site has less bare soil and more surface litter cover than both the sagebrush and woodland sites. Vegetation at the mastication site is primarily herbaceous, and the site has 50% greater total foliar cover than the woodland. Hydrology and erosion results are in process, but we observed greater flow dispersion through the thick cover at the mastication site relative to the more bare intercanopies at the woodland site. Detailed results from this study are expected in late 2025. The unique data set and process understanding acquired in the SageStep study summarized today has contributed to various synthesis products on the effects of woodland encroachment and woodland ecohydrology and to advances in a suite of hydrology and erosion models as depicted in the slide. The work at Grand Staircase-Escalante National Monument will contribute additional data to this effort and understanding for these systems and will aid land managers in predicting responses to management practices. We will continue to apply our methodologies to different ecological sites and across treatment types to advance current understanding. Thank you for your time today.